Good morning everyone. Welcome to All 4C learning classes, session 13. Let's have some recap of what we have learned yesterday. That is, the digestion results in the formation of food bolus in the buccal cavity, then the chyme in the stomach and chyle in the intestinal region. Basically, our digestion start with the carbohydrates. Next, the food bolus that enters into the stomach. In the stomach, protein digestion takes place. That is, the gastric juice that are added to the food results in the digestion. This digestion of the proteins by pepsin. There are the two hormones that acts under the acidic medium is the pepsin and the renin. Renin in the infants, whereas the pepsin, renin only in the infants, whereas pepsin and the renin in infants, but only pepsin in the adult individual. Proteins are digested under the acidic medium in the stomach, and renin is artificially synthesized in the form of renites. Renite is a commercially product extracted from the gastric cough, that is the gastric mucosa of the cough. And there are certain times when the food from the stomach squeezed into the esophagus results in the burning sensation, which is named to be the heartburn or pyrosis or hyperacidity, which is nothing to do with the heart problem. The food that is taken into the mouth, then into the buccal cavity, that is which moves, which is named to be the swallowing or the deglutition. The failure in the deglutition is named to be the dysphagia. And the different types of the enzymes in small intestine. The partly digested acidic food in the stomach is the chyme, which moves into the intestine. In the intestine, it is undergo the complete digestion. Carbohydrates into monosaccharides, proteins into amino acids, fats into fatty acids or fatty acids and glycerol. Besides that, the nucleic acids are also digested by means of the nucleases, the enzymes that the influence in the intestine is carbohydrases, then the proteases, lipases, nucleases, nucleotidases, nucleosidases. This is all what we have seen earlier. And the digestion is influenced by the neural and hormonal control. Digestive activity is under the control of neurons and the hormonal activity. Here where we discuss the neural control. Neural control is nothing but the endo or intrinsic neural activity, as well as the extrinsic neural activity, What I told you is the neural activity and the hormonal activity influences the digestion process. The neural activity discussed under intrinsic neural activity as well as extrinsic neural activity. What do you mean by intrinsic and extrinsic? This is by means of the nerve plexus. Nerve plexus we have discussed. The nerve plexus are the network of the nerve fibers as well as the parasympathetic and sympathetic nerve fibers. Combination is nerve plexus. They are what present in the muscular layer of the wall of the gut, and between the muscular layer and mucosa, that is in the submucosa, which is present in the wall of the gut, so it is intrinsic, internal. Whereas the extrinsic or the neural activity, this activity called the intrinsic neural activity, what causes the peristaltic movement and the gastric juice release. Whereas the extrinsic activity modifies the activity of intrinsic, which means these two together what influence the digestive activity of gastrointestinal tract. By this we come to know that not only the chemical digestion, microbial and the mechanical digestion, there is an activity required from the neural control. That is the neural activity and hormonal activity. However, the neurons and hormones are what are said to be the controlling and the coordination, one by the nerves and one by the chemicals, that is hormones. So in this connection, we learn the intrinsic neural activity and extrinsic neural activity, the one that is within the gut wall. We have seen the histology of the gut, where we see the serosa, muscular layer, submucosa and mucosa. In the case of the what is called a muscular layer, the network of the nerve fibers with that of the parasympathetic neurons is what named to be the Erbox plexus, and that present in the submucosa, the network of nerves with that of the sympathetic neurons is inside the wall, name it to be the submucosal plexus or Messenger's plexus. So the network of parasympathetic nerves and nerve fibers is Erbox plexus or the mesenteric plexus, which is responsible for the controlling the peristaltic movement. Do you agree that the peristaltic movement is one of the digestion process? Definitely. Gastric mobility is also needed for the digestion process. So here is this become an evidence? Definitely. The gastric movement causes the secretion of the gastric juice and also the movement of the food in a wave-like manner from one part to the another. This indicates that the neural activity and the hormonal activity influences the digestion. That is by intrinsic neural activity by nerve plexus, which is the Erbox plexus and the Messenger's plexus. Messenger's plexus. Next, the extrinsic, which is from the part of the nervous system, that is sympathetic and parasympathetic neurons, which innervates into the gut and influence the gastric activity, since it is outside, that is extrinsic. Extrinsic neural activity which modifies the activity of the intrinsic neural activity. This is what the neural regulation activity in the digestion process. Certain times you might have experienced the smell of the food, the sight of the food. When somebody is preparing the best food, the delicious food nearby you, immediately the saliva is secreted, which means the sense of sight, sense of smell, what act as a stimulation. This stimulation is what regulated by, this is what done by vagus nerve. Vagus nerve, the 10th cranial nerve. When one exposed to the sense of the smell of the food, sense of smell of the food and the sight of the food, The vagus nerve is stimulated and send the signals to secrete the saliva. This is the main region. This is the summary behind our secretion of saliva. When you have a sight of the food and smell, and this is how we see the smell and sight of the food also influences the secretion of the juices called the salivary juice, which is the stimulus is received by the vagus nerve and release the salivary juice. We do take food at a regular interval, which is the prime reason for the good health. When at a particular time when you take the food, the normal activity takes place. So at a particular time, as you are taking the food, so you get the hunger. So when at a particular time you get the hunger, it is nothing but the conditional reflex. That is, at a regular interval you take the food, so our body responds. That's what conditional reflex. At a particular time one gets the hunger, it is nothing but the conditional reflex. This is what all we see in the case of the digestion process. Let's come to the discussion, that is absorption. What do you mean by this? We have discussed that the digestive system is for digestion. Digested food is absorbed. The unabsorbed food is what defecated to the outside, that is, it is eliminated, expelled outside, which is named to be the egestion. So our digestion system perform the four functions, which is ingestion, which is done by mouth and the buccal cavity and the pharynx. Next, digestion done in the buccal cavity, oral cavity, then in the stomach, and it is completed in the small intestine. Now the digested food needs to be absorbed. This is what are our content of the topic today. Absorption. Absorption is the digested food is what diffuses into the blood and lymph. The process of diffusion of the digested food material called proteins, end products, amino acids, carbohydrate end products, monosaccharides, glucose, fructose, galactose. Next, fats end product, fatty acid, glycerol, nucleic acid end product, pentose sugar and nitrogenous bases. These are all what the simplified substances which are diffused into the blood and the lymph. That is what absorption, the process of the diffusion of completely digested food materials called the fatty acid glycerol of the fats and the monosaccharides of the carbohydrates called sugar, fructose and galactose. Next, the nucleic acid and the products are the pentose sugar as well as the nitrogenous base. Proteins and the products, amino acids are diffused into the intestine, which is through the villi. Villi increase the surface area of absorption. Each villi is a finger-like projection. Villi contain the capillaries of the artery, vein and lacteals. So this is what the absorption. In this absorption process, the fats are they are insoluble in water. The point to remember, the villi increases the absorption. That is, they are responsible for increasing the surface area. Basically, the fat is insoluble in water. Blood is with what we are discussing is that the diffusion of the end products of the digestion, simplified substances into the blood and lymph. Why lymph when it is said the blood? Generally, the blood is with the plasma and the blood cells. Plasma contains 92% of water, but fat is insoluble in water. So how the fat enter into the blood directly? So it requires some accessory route that is none other than lymph. To remember this. So this absorption is the process which is takes place by simple diffusion, facilitated diffusion, active transport and osmosis. Simple diffusion, the diffusion of the substances by what is called a concentration gradient. Generally, the simple diffusion takes place. The glucose, amino acids and the chloride ions are absorbed. Simple diffusion, the diffusion of the substances by concentration gradient is what named to be the simple diffusion. Facilitated diffusion, the diffusion of the substances may or may not be with the concentration gradient, and which is facility. What do you mean by facility? Something help. So absorption of the nutrients help with the help of the carrier proteins. What we name it as the facilitated diffusion means the absorption of, we see the glucose and amino acids. Glucose and amino acids are absorbed with the help of the carrier proteins is what we name it as a facilitated diffusion. Now you got my point, the difference between that simple diffusion and the facilitated diffusion. The diffusion, if it is a help or facilitated by the carrier proteins, we term it as facilitated transport or diffusion. Next, come to the active transport. The diffusion of the substances against concentration gradient, which requires the energy, is what active transport. Basically, glucose, amino acids, sodium are absorbed by active transport, and osmosis, that is the water absorption is by osmosis. That is generally water content we require more, so it is continuously absorbed. Why means we know very well a cell which includes the cytoplasm. 60% of the cell is with the high content of water. This is the prime reason why a person died due to dehydration, but do not die lack of food. A person can survive without food but cannot survive without water. Why? The dehydration kills a person. So the absorption is the process of diffusion of the digested food materials into the blood and the lymph is what name it to be the absorption. This absorption is of In human beings, absorption is done by means of simple diffusion, facilitated diffusion or transport, active transport and osmosis. If the absorption of the food to the concentration gradient as per the concentration gradient is named to be the simple diffusion. That is, glucose and amino acids are by simple diffusion. Next, facilitated diffusion or facilitated transport. The diffusion of the substances which is facilitated by carrier proteins is what named to be the facilitated transport or facilitated diffusion. This is the glucose and amino acids are comes under the facilitated diffusion transport. Next, active transport. Active transport is the transportation, the diffusion of the substances, the simplified substances against the concentration gradient, which is what by energy. The diffusion of the substances which is by energy consumption or by energy is active transport, which is against concentration gradient, which means it is from lower concentration to the higher concentration. It is against concentration gradient. So here it is concentration gradient means from high to low naturally takes place, but it is against that which is by energy. That is active transport to glucose, amino acids and sodium is active transport. And osmosis, the absorption of the water is by osmosis itself. So this is how we see the absorption. So end products of proteins are amino acids. End products of the carbohydrates are glucose. End products of basically nitrogenous bases, which includes the amines. So furtherly they are break down into the proteins, and that is nucleic acid end products. Whereas what is not taking place here, that is fats. End products of fats are fatty acids and glycerol. So all these substances are what absorbed into the blood capillaries. Since they are smaller molecular weight and are what is called soluble in water, but there is no point to be discussed about the fatty acids and glycerol. Then the fats are with a short chain of fatty acids, and a long chain of fatty acids. Short chain are with a smaller in size, so they are absorbed. But mostly the long chain of fatty acids are present, which are with a large molecular weight, which are larger, we say. Since they are larger and have no solubility in water, so they are not directly diffused into the blood. Then they require some other activity. So simple diffusion, facilitated transport, active transport and osmosis. Now the thing is to discuss how about the fats. So we discussed that fat are large molecules and insoluble in water. So they require some other activity of the diffusion, what is called absorption. The fats, as I told you, the short chain of fatty acids and the long chain of fatty acids. Now the fat is in the intestinal cavity, that is in the cavity of the gut. In the cavity of the gut, the emulsification takes place. Already we discussed that the emulsification is the process. Emulsification Emulsification in the sense the conversion of larger molecules of the fats into smaller molecules of the fats. So this emulsified fats, which are the droplets, the emulsified fats which are droplet like, is what name it to be the micellase. So micellase formation takes place in the lumen of intestine itself, lumen of intestine. So the absorption of the fats require not the same process what we see earlier. So it requires some accessory route, that is nothing but first of all in the lumen of the intestine where the emulsification of the fats takes place. This emulsification of the fats is named to be the micellase. Micellase are none other than the droplets of the substances, the droplets of the fat molecules which are smaller in size. Nothing but the emulsified fats, name it to be the small droplets of fat molecules, are named to be the micellase. These micellase are formed in the lumen of intestine. This micellase that are formed in the lumen of intestine, first absorbed into the epithelium. Micellase are absorbed into the epithelial cells. We do discuss that the alimentary canal is lined by means of columnar epithelium. So the micellase are absorbed into the epithelium. In the epithelium of the villi, that is in the intestine, these are what covered by means of protein coated fat substance, name it to be the chylomicrons, that is the micellase are emulsified fats, nothing but the small droplets of fat molecules is nothing but the products of the emulsification. We discussed that the fats are not at all digested until and unless emulsification takes place. This emulsification results in the formation of the small droplets of fat molecules, micellase, in the lumen of intestine. Then they are diffused into the epithelial cells. In the epithelial cells, endoplasmic reticulum is present. Where with the help of the endoplasmic reticulum, the micellase are what converted into the protein coated fat substances or fat molecules, name it to be the chylomicrons. Now these chylomicrons are the protein coated fat globules are released into the lymphatic duct, which is named to be the lacteals. Look at here, this is the diagram of a lacteal. In the sense what do we term lacteals is a lymphatic capillary. So the micellase formed in the lumen of the intestine are diffused into the epithelium. In the epithelium, by the action of the endoplasmic reticulum in the epithelial cells, or what forms into chylomicrons. These chylomicrons that are formed in the epithelium of the intestine by exocytosis. Exocytosis, in the sense exo means outside, cyton means cell. So by exocytosis, the chylomicrons are diffused into the lacteal duct, which is the lacteals. Lymph capillaries name it to be the lacteal duct. It is water with a necessary route. Basically, we do require a necessary route. The capillaries in which the food are diffused into the extracellular fluid. From the extracellular fluid, there forms the minute capillaries, name it to be the lymph capillaries. So, the lymph capillaries or lacteal duct is for the transport of the fats. Why? As they are insoluble in water and larger, so they are not diffused into the blood but have a accessory route called the lacteal duct, lymph capillaries. This lymph capillary, what opens into, furtherly continued into lymphatic duct, thoracic duct, then through subclavian vein. Subclavian vein, it releases into the blood circulation called vein, which means it is a necessary route from the extracellular fluid, that is the fluid between the cells of the tissues, into the lymph capillaries, then into the lymphatic duct, thoracic duct and subclavian vein, then into the bloodstream. This is how the fat absorption and the transport takes place. While this transport, the endothelial wall, endothelium in the sense, what we learn is the epithelium lining the blood vessel. The inner wall of the blood vessels is with the simple squamous, name it to be the endothelium. The endothelial walls of the blood vessels release lipoprotein lipase. Lipoprotein lipase is what we see secreted from the endothelium of the blood vessels, is what act upon the chylomicrons and convert them into the what is called triglycerides. They are what break down, and from there they are, by the action of lipoprotein lipase on chylomicrons, the end products are absorbed and reach the adipocytes of the liver and other tissues where it is stored. So the point you remember, the fat is stored in our body parts, that is at the adipocytes of the liver and other parts. This is how it reaches. This is what we learn the absorption process. In this absorption process, diagrammatically see, we have seen the diagram gut, that is in the stomach. The substances that are absorbed in the stomach, diagrammatically you can see in the stomach, water. The functions of the stomach we have seen: water, alcohol, lipid soluble drugs, and apart from that, simple sugars and water are absorbed in the stomach. Even the drugs we say is aspirin. Aspirin is a drug, medicine. So in stomach, the water, simple sugars, that is monosaccharides, alcohol, aspirin are what absorbed. Next, in the duodenum is the part where the iron, calcium and amino acids takes place. This iron absorption is facilitated by causal intrinsic factor. Causal intrinsic factor is one of the factor produced in the stomach. which play a role in the absorption of iron. Thereby it is in turn helps in the erythropoiesis, the production of the WBCs. So causal intrinsic factor is indirectly help in the what is called hemopoiesis. Then in the jejunum is a part of the small intestine. We learn that most of the food is absorbed in the small intestine. So jejunum were monosaccharides, glucose, fructose, galactose, fatty acids, some extent. That is, we are not discussing whether it is into the blood or lymph. That's what jejunum, monosaccharides, fatty acids, glycerol, amino acids, vitamin and water. Then in the ileum, the cyanocobalamin, vitamin B12, and bile salts, water, colon, drugs, water, minerals and vitamins. By this you come to know the absorption of the drugs is in the mouth as well as in the stomach and large intestinal parts.